400, but as I mentioned, there are two different batches of slides. If yours looks more purple than red or bluish, it kind of has a different appearance. And I'll look at both of these. I'll show you both of these on the demo here. If we look at, if we zoom a little bit over, you see this one also has some mature sperm, not quite as late a slide as this, but whenever they look, the stain, the dark stain is the DNA. And so when there looks like these sort of little lines, that means you're getting to mature sperm. These big ones, with a lot of chromosomes, you can see these very large cells, those are the primary spermatocytes. I won't ask you to identify every cell in here. Sertoli cells, as I said, they're hard to find. I won't ask you to identify them, but you should certainly identify Leydig cells outside the seminiferous tubule, the spermatogonia, which are the round cells near the periphery, primary spermatocytes, which are very large and with distinct chromosomes, and then mature sperm, which are sort of dash-shaped and darkly stained.
Which ones were the sperm?
What's that?
What was the first one?
What is the first one?
Yeah.
Spermatogonia. Those are these round ones, these sort of smallish round cells near the periphery. There's also spermatids, as we'll see, are easy to spot too. He said confidently. Maybe not in this particular preparation. Yeah, the spermatids are not showing up very well in this one. But you can see these bunches of Leydig cells in between. That in this preparation, the nuclei is stained very dark. What they are is the two different slides are actually stained different. And so they'll have different appearances. This is another one with a lot of mature sperm. But you see, because of that spermatogenic wave and the staggered arrangement within the seminiferous tubules, some will look like this, while the one next door may look like that and have no mature sperm visible in it. Right? This one, by the way, this one is loaded with spermatids. The spermatids are these smallest cells, very lightly stained. Right? Looks like a whole bunch of eggs in there. Right? Looks a lot different than those other sections that I showed you. So spermatids are very easy to spot. Spermatogonia are easy to spot. Mature sperm are easy to spot, and primary spermatocytes are easy to spot, and Leydig cells. But don't worry about secondary spermatocytes and Sertoli cells. You should know what they are and where they fit in the sequence, but identifying them in the slide is tricky, and I won't ask you to do it. That again has a lot of spermatids, a lot of spermatids in this one too, a whole lot of spermatids, and then this one has got a lot of mature sperm, almost mature, and then we had this one where you have loads of them ready to, just being sloughed off there. A whole lot of mature sperm in that one too. Okay, now I want to take an example of one of the other batches of cells.